Um, Hello, everyone. Thank you for the introduction. It's such a great pleasure to be here in person. I'm actually a postdoc at Dr. Mario Ostrowski's lab at the University of Toronto. So today I'll be talking about immune response to COVID-19 vaccines in older people living with HIV and effects on HIV reservoir. In our study, we aim to understand if people living with HIV aged 55 and over have similar or diminished immune responses to COVID-19 vaccines compared to HIV negative controls, how these responses might change with each dose of the vaccine, and how different they might be in different subgroups, such as immunological responders and non-responders, and whether or not COVID-19 vaccines might adversely affect HIV reservoirs. So the start of this study about 2 years ago, we hypothesized that COVID-19 vaccines will adversely, will induce a diminished immune response in people living with HIV in terms of durability and robustness, and will adversely affect HIV reservoir. We recruited 91 participants for our study. We're actually very lucky to have recruited 57 with the baseline, and why lucky because the vaccines, COVID-19 vaccines, were rolled out very rapidly in Ontario for this particular demographic. So we were indeed very lucky to sample the baseline for 57 of them. So here on the left, you can see the share of different subgroups based on their HIV status. So we had 23 HIV negative participants, 42 immunological responders, 20 non-responders, 5 low-level viremics, and 1 long-term non-progressor. Our HIV group and the negative controls were age-matched. The HIV negative group, the median age for it was 62, and for the HIV group, it was 63. It's a longitudinal study, so this is what our protocol, our timeline looks like. You can see here the 3 doses of the vaccine shown in red, and we did have to sort of introduce quite a number of changes, to be honest, because the procedures and the rules were evolving, changing all the time. So we had to add some additional visits over time. But you can see visit 1 here shown in blue is the baseline. Our first main time point, it used to look green now, I think it's more like yellow. Visit 8, I guess green or yellow, which is 24 weeks after the first dose of the vaccine, which is also a half a year mark. And the second main time point, and the last study visit, which is visit 9, it's 48 weeks after the first dose, which is also 1 year mark. So our endpoints included live virus neutralization titers in serum at week 24 and 48. We also looked at serum IgG levels to spike RBD and also NP, as well as at salivary IgA and IgG levels. However, I'm not going to talk about this particular part today because it's still being finalized at the moment. Also looked at T cell responses against spike, but also GAG and NEF, although for different reasons, at baseline week 24 and 48. And of course, we're looking at the size of the HIV reservoir, for which we calculated the frequency of intact proviruses in CD4 T cells, again at baseline, week 24 and 48, and we did some other tests as well on memory B cells, for example. So on this plot, you can see our data for RBD-specific IgG levels in serum. So you could just make more sense of what you're looking at for each time point here. You can see that it's split into 2 parts by vertical dashed lines. On the left, we are comparing the bulk of HIV samples shown in green to the HIV negative participants shown in blue, whereas on the right, it's a breakdown essentially where we show separately immunological responses shown in turquoise and non-responders shown in purple. So you can see here, of course, that with each vaccine dose, the level of IgGs increases, then it begins to sort of decay gradually until the next dose is administered. And then it goes down to even a higher level. And so the highest level is achieved after the 3rd dose. Importantly, we did not observe any significant difference between the HIV group and HIV negative controls, with one notable exception, which is at visit 8B. We saw a significantly higher response in the immune responders group compared to the HIV negative controls. So here we have live virus neutralization titers, and here the results have, in contrast to what we saw before. We saw much lower neutralization in the HIV group at week 24 compared to the HIV negative controls, but this difference disappears by week 48. Why that might be, well, we could guess that that's due to this discordance could be because of, could be related to epitopes outside of the RBD domain, perhaps. We also looked at the frequency of RBD and antigen-specific memory B cells. This work was performed in Susan Moore's lab at NIH. We did have to go with a much lower sample size here because the B cell work requires, it has a lot of stringent criteria that these samples had to match. So we picked 17 samples and analyzed them, but essentially you can see the same trend here as in the neutralization data that from the previous slide. We saw a gradual increase in the frequency of B cells first at 24 weeks and then at 48 weeks. And then we looked at the correlation of these frequencies with the neutralization titers. We only saw a moderate correlation, which wasn't statistically significant, but that's probably due to the smaller sample size. So we also decided to perform an RBD surrogate neutralization assay in order to see what exactly we're looking at with the live virus neutralization assay, how much it would be related to RBD. Here we added 2 additional time points that would be closer to their respective vaccine doses. But if you look at week 24 and week 48, you can see this pattern is essentially the same as in the live virus neutralization. Only the responses at week 24 are lower, which is, again, as you can see from the previous time point, it's the result of a decay of neutralizing antibodies. But other than that, the pattern is essentially reproduced here. Another important thing that I would like to draw attention to is the difference between immunological responders and non-responders. The response was higher in the former. So we also looked at the correlation between these titers and the live virus neutralization titers, and indeed saw quite a strong correlation at both time points, both at visit 8 and visit 9. Speaking of spike-specific T cell responses, I'll just first explain what exactly you're looking at, because there's a lot to unpack. So the left and the right panels are essentially the same. On the right, on the left, we're showing the difference between the bulk HIV samples compared to the shown in green, compared to the HIV negative controls shown in blue, whereas on the right, it's essentially the same, but with a breakdown where we show immunological responders and non-responders separately. So we stimulated our cells with different peptide pools, with S1 peptide pool, S2, and a master spike pool. And then we looked at interferon gamma, IL-2, and dual responses. And for each scenario, we're presenting 3 different time points: the baseline, 24 weeks, and 48 weeks. So we saw the results were more resolved for IL-2, but basically the main conclusion that you can make from these results is that the responses were either very similar in the HIV group or were actually higher than in the HIV negative controls. So if you look on the right, you will see that this difference is mainly due to higher responses in the immunological responders group. They did mount a stronger response but both compared to the non-responders, but also interestingly enough, when compared to HIV negative controls. So we believe that could be due to potentially an imprinting of for Th1 response in the HIV group, because we know that COVID-19 vaccines, they predominantly elicit Th1 response. And of course, in the HIV group, due to chronic infection, HIV infection, they could potentially mount a stronger Th1 response, particularly in the responders group who have preserved immune system. We also looked, speaking of potential effects of COVID-19 vaccines on the HIV reservoir, we also looked at GAG and NEF-specific T cells, but we did not see any difference when compared to the baseline. So both at 24 weeks and 48 weeks after the first dose, their frequency was the same as at baseline. And of course, we also looked at the frequency of intact HIV proviruses as the measure of the size of HIV reservoir. So we specifically looked at the frequency of intact proviruses in CD4 T cells at week 24 and 48, and compared them to the baseline, and we saw no difference, which means that these vaccines, they did not cause an increase, with one notable exception of low-level viremics, although we only had 3 of them. But as you can see at visit 9, the last study visit, they all had approximately 60% increase in their intact HIV DNA compared to the baseline. And finally, speaking of frequency of breakthrough COVID-19 infection, which is the measure of vaccine efficiency, we also didn't see any evidence that there would be lower efficiency in the HIV group. Quite the opposite, actually. We saw higher proportion of breakthrough cases in the HIV negative group, but that difference wasn't significant because we didn't have the sample size to generate a significant conclusion from this. So just to recap, so we saw that people living with HIV and HIV negative controls aged 55 and over generated similar levels of spike and RBD IgGs in response to COVID-19 vaccines, and these levels increased after boosting in all groups and being the highest after the 3rd dose, which tells us that the 3rd dose was really of huge benefit. We detected less neutralizing antibodies in people living with HIV than in the negative controls after 2 doses, but that difference disappeared after the 3rd dose. Again, another evidence for the benefit of the 3rd dose. We saw similar or stronger spike-specific T cell responses in the HIV group compared to HIV negative controls, both at 6 months and 1 year. And we found no evidence of adverse effects of COVID-19 vaccines on the size of HIV, on the stability of HIV reservoir. And again, no evidence of low efficiency of COVID-19 vaccines in older people living with HIV. I would like to thank people who contributed to this project. It's a collaboration, and some of our collaborators here today, including Colin Kovacs and Susan Moyer, Tawak Chun, and Rupert Hall. And thank you for your attention.